Hello, everyone. I'm Dr. Carmen Pugliafito, and welcome to Retina Synthesis. Today, we have with us Professor Jose Sahel of the University of Pittsburgh Medical Center, where he is Distinguished Professor and Director of the Eye Center. Dr. Sahel was recently awarded the International Prize for Translational Neuroscience for his pioneering work on optogenetics for retinal degenerations, the topic of our today's session. Jose, welcome to Retina Synthesis.
Thank you, Carmen.
So tell us what the concept of optogenetics is.
Yeah, so optogenetics is some mechanism that was identified in very elementary organisms like algae, enabling them to detect the light and to respond to light. These are proteins that are both sensitive to light, capturing the light and triggering a change in the membrane potential, which is eliciting a response to light. And this type of proteins do recapitulate into one single protein what is happening at the level of photoreceptors in the human retina. And in the human retina and mammalian retina, you need several proteins, sometimes hundreds of proteins to do that. So we thought, like a few of us, but this is a very nice way to activate photoreceptors like any type of neurons by this type of proteins, by expressing them. This is used as a tool in neuroscience by many laboratories across the world as a way to manipulate neuronal activity in any cell type. But what we thought with Boton Roska, my collaborator, and my team in Paris at the Vision Institute was that we could use that as a mechanism to activate the remaining cells in the retina. So in retinitis pigmentosa, for example, the photoreceptors are dying, so there is no response to light. And the goal is to activate the remaining cells in the retina, for example, the ganglion cells. And this is what is called optogenetics, activating by light using genetically encoded proteins.
So how do you deliver the pigment, the
optogene?
material?
So, we use gene therapy. So we use a vector, and this is done in our case using an intravitreal injection, and the protein is then expressed in the remaining ganglion cells in the retina. So it's a combination of a gene therapy to express the protein, and then you need to activate the protein with light, and we use a specific device that we developed, goggles that are activating in the specific wavelength that is required for the activation of a protein.
What's the wavelength?
So it's in the amber light, it's red to amber, and this wavelength is not toxic, it's not very energetic, so you can use it without reaching any threshold for light toxicity.
So the patient wears a special set of spectacles?
Yeah, that's right. Yeah, this is a difference as compared to other groups that are trying not to have spectacles, but we believe that this is better because you need to adapt to many levels of lighting. In the natural environment, we are using many levels of light from very dark to very bright, which is in the range of 11 log units of light intensity. And there is no protein that can adapt to that range of lighting. So the goggles are doing that, the spectacles are adapting to the levels of lighting, and this is stimulating in the right wavelength at the right intensity.
So what was the results of the clinical trial?
So the clinical trial is still ongoing. We currently have treated like 10 patients. It was a dose escalation trial. We started with a low dose and then mid dose and then higher dose. And because it was very safe with no significant inflammation, now there is an extension cohort that is getting the higher dose of a vector. So the patient that we reported previously in Nature Medicine a couple of years ago was in the first cohort, and he was treated just before COVID, and he was able to start the rehabilitation process, to start to use the goggles, and he spontaneously started to report that he was seeing the crosswalks, the stripes. And then we brought him to do more testing, and he was able to find objects, to identify objects, and to grab objects, and to quantify how many objects were on the table. And now, in the meantime, this patient has come back several times, but we have also been able to do that on other patients. Now, the study is still ongoing, and several patients in the study have been reporting the same outcomes. We are actually compiling the data from this first group of patients to publish a new paper to really demonstrate the efficiency that we were able to achieve. Importantly, because we wanted to check that the responses that the patient was describing were reliable, we are using also recording at the brain level using the multi-electrode electroencephalography to monitor the light responses at the cortical level. And it corresponds very well with what the patient is doing. When the patient is perceiving any object, there is a specific change in the oscillation at the occipital level.
So that's impressive. So there's a correlation, a neuropsychological correlation.
Yeah, exactly. And now we have not yet reported on that, but there is an encoding system that was developed using mathematical algorithm to correlate the shape of the object with the recording that is done in the brain. And this is very consistent too.
So this would be acceptable for all forms of widespread retinal degenerations.
As long as you have optic nerve functional, as long as you have ganglion cells remaining in the retina, you could use that to activate. Of course, as of today, the resolution is not very good. It's just seeing objects and locating objects, but we are working on refining actually the technology using a goggle stimulation to find a way to improve the visual acuity, but we are not yet there. As you know, there are other groups. I think you did interview a couple of groups that are working on other technologies that also could be working. What we believe is that the combination of the gene therapy and the goggles is a good combination, but of course, this is early stages of this research and more to come.
Is there any chance of changing the wavelength that's involved or not?
So this wavelength is the best, actually. Many groups are using a green or blue light, and this is toxic because you are the efficiency, the light efficiency, and the range of toxicity is higher. So this is optimized already to be in the red is a better wavelength, and this is what we believe is probably going to work better. What might happen is to enhance the sensitivity, and there are a few groups that are currently developing that to enhance the sensitivity so that you may even not need the goggles or need lower levels of light for activation. So we are interested in seeing what it does. I still believe that you need to adapt to so many levels of lighting, but I don't think you can eliminate totally the goggles or the spectacles.
Are the goggles active optically in terms of modulating the light intensity?
Yeah, they are doing that. They respond to any levels of lighting and they average that. So it does project in the right wavelength and the right intensity of light. And the goggles are responding every pixel to any light intensity. So they are very efficient.
So what's the future of this?
So the future is currently we are working on optimizing the specificity of the cell transfection. Currently, we are activating any cell type. It's a bit similar to what was done with retinal implants. So we are trying to see if we can be cell specific. And we are also working on the algorithm of stimulations, which means that even the patients that were in the existing trials could benefit from it because it's more or less like computational algorithms that are going to compute the information and send to the right wavelength and more importantly, the right signal. So we are working on that currently to optimize the signal.
So what are the glasses connected to?
So they are just sending the light onto the eye. So they are wireless glasses that are the patient is wearing that on the head, and they have a camera in front of that and the projector at the back of the glasses sending into the eye. But there is no specific connection. They are just wireless glasses.
So that's fantastic. Are the clinical trials being conducted in the United States or in France?
Yes, so it's done currently in Paris and in Pittsburgh. And the plan for the next phase of the trial is to open a few more sites, both in the US and in Europe.
So have you concluded on the right dose of the vector?
Currently, the higher dose is very well tolerated, so we believe this is the right one that we are using.
That's exciting. So
It is.
Thank you so much for this interview, and congratulations once again on winning the International Award on Translational Neuroscience. I know you've been working on this for many years.
Yeah, with Boton Rosca, we got the award together. Boton is in Basel, and we have been pairing the work together, and my team in Paris at the Vision Institute, and now my team in Pittsburgh.
Okay, wonderful. Thank you so much.
Thank you very much. Bye-bye.